Hi friends, welcome back to the JK Biology, and today our discussion would be on molecular markers. That's how molecular markers is used to genome mappings and how it becomes identified. Okay, so without lapsing times, I will be describe that types of the molecular markers. That is two types of molecular marker, that is isozymes and second is DNA sequences. Okay. The most molecular markers used is the DNA sequences for the chromosomal mappings, and they may be also two types, that is defined. First is defined and second is anonymous. Okay, the anonymous molecular markers means that's not known specific function and specific features of that sequences. That is anonymous marker. That means the sequences of that genomes, that sequences is not known of that specific position and specific feature and also of its function, but the defined markers have already known that their specific functions and features. Okay, like restriction digestions, rare cutting restriction enzymes, that is already defined. That's where the enzymes would be cut in the sequences, but also another features can be known of the defined markers. The defined marker may be maximally becomes polymorphic. What is the means of the polymorphic? Today you can see here also that's the means of polymorphic. The defined marker becomes polymorphic. Polymorphic markers, and for examples RFLP, that is restriction fragment length polymorphism. Okay, RAPD, random amplified polymorphic DNA sequences. Okay, and STS, sequence tag sites, and also AFLP, amplified fragment length polymorphism. That is the polymorphic DNA sequences are polymorphic markers, that is also defined markers. Okay, now come to the points that why say it is called polymorphic. Because suppose that's our genomes, our DNA sequences differ from you, from people's. Why I am looking different from you, from another person's, our face, our skins, and also our bodies. Why? Because the changes in the sequences from individual to individuals, that is why it is in the sequences. So these regions, that suppose that we are taking restriction fragment length polymorphism, that's restriction enzymes is used. Why? Restriction endonuclease, you know that's restriction endonuclease is that there is the sequences. Suppose that I have sequences as like A T C A C C A C G. That is the sequences. That is 9 sequences. I am taking simple examples. That is the 9 sequences. This is 9 sequences. Okay. Restriction fragment, restriction endonucleases. That means the enzymes that is restricted to cutting the sites in the specific position of the DNA sequences or the genomic sequences. That's how restricted sites. That means that cannot randomly cut in anywhere. That means there is the enzyme, that is the structure. Suppose that we are taking the exact structures of the restriction endonucleases. Endonucleases, why say called endonucleases? Because within the DNA sequences that's cut in the specific position, that's not outer sides. That is called exonucleases. That's remove the outer side of the sequences. That is called exonucleases. But that is the endonucleases that cut within the DNA sequences. Okay. Suppose that this is the structure of the restriction enzymes that's recognize A, T, C, that's fit in that specific position. Okay, so why this restriction endonucleases binds to that position? We are taking that's not 3 sequences, we are taking only simple T. Okay, so this T are binds to the A. For taking example, not take seriously, that's A bind to the T, and so these regions they cut to that positions. and also our, that is the single standard I am showing. This becomes double standard, that is A, G, T, G, G, T, G and C. This is the double standard sequences of my, this is mine sequences for example. So that's this restriction endonuclease recognize to that's okay and cut to that position for taking, that's cut this RE cut to that specific positions. Okay, so what would be the result? That is A sequences and is removed and also T, C, A, C, C, A, C, G, that is the sequences, and this another sequences T, A, G, T, and there is also cut into G, G, T, G, C. Okay, so that's looking like this. Okay, looking like this, for example, we are taking. Okay, and long sequences that's looking like as this for longer sequences. Okay, so that is also called the sticky ends because that is the regions, that is edges. So this region that is called the sticky ends. When cuts in equal proportions, that's there is not a sticky end that is equal here, that is called the blunt end. For I will describe in another lectures. So this is called the sticky ends restriction endonucleases. Suppose that's eco R1, that is the many different types of the restriction endonucleases found has been identified. So we are taking eco R1, that is the restriction endonuclease, cut that specific positions, that is mine sequences. Okay, cut in our genomic DNA where the T sequences, that is the TGTAC sequences, 4 sequence that's recognize the 4 sequences AGTA. For simple examples, not I am taking that exact of eco R1 that recognize in any base sequences, but for example you look here, that's they recognize if that sequences is found in these sequences. Okay, AGTA that is not mentioned, but for usually understands that you will take care that TCAC. Okay, so where these sequences in our genomes becomes very long, very bigger. That's not a limited sequence. Many sequences is found in many positions. That sequences is found. So where this sequences is found, so restriction endonuclease cut to that sequences. Suppose in after then there is also ACAC sequences, TCAC sequences is found in another positions for the long genome. That's there is single cuts, and if after several sequences that sequence is found, restriction endonuclease recognize that position and cut the sequences. So different fragments of the length of the DNA is found from single restriction endonuclease in mine genome, that is mine genomes. Okay, so there is a, suppose that's that types of sequences is found in 5 in numbers in mine sequences. So what would be the results? 6 fragments would be found, that is 6 fragments would be found in my mine genome sequence. Okay, mine genome sequence for easily understand. So okay, not take serious. 6 sequences is found from that restriction endonucleases. Okay, then in another individuals, that is that single mutations or single deletion or addition of the sequences can change the whole sequences of use of another people's. Why? Because you can take example for simple, that's there is a deletions of the T sequences. Always that the sequences is read in triplet forms, that is triplet codons, that is 3, 3 and 3. 3 codons code for a single amino acids. Okay, that is the triplet of the codons. So if there is the mutations, that is means deletion. So what would be the results? A, C, CCACG, single mutation. So the triplet codon read as look like this. You can check here. Look like this. The sequence becomes changes. The sequence becomes changes in another individuals. So looking that's individuals different than me. Okay. So that changes. What would be the results? That's T, G, T, G, G, T, and GC. That's sequence of that individual. So the sequence is changes. The restriction endonuclease, if A, T, C, A, C recognized in that sequences, then the cuts of that position. But there is not that sequences is found T, C, A, C. You can see here that there is no T, C, A, C. So that restriction endonuclease does not recognize on that sequences in that specific positions. I am taking examples in that specific positions. Okay. So that's not recognized in another individuals. Okay. So why is it called? This is the polymorphic markers that's differ in individual to individuals within population. Okay. Differ. So this becomes called the polymorphic DNA markers. I was taking the restriction endonuclease of the RFLP, restriction fragment length polymorphism. That is what the examples where restriction enzyme was used. Also, I am taking the examples of RAPD. Okay, RAPD that does not require the restriction endonucleases. Okay, that does not require restriction endonucleases because there is a sequences. Suppose that this is the sequences of mine where for RAPD a primer is designed, that is synthetically primer is designed. The primer sequences is known. That's become 10 nucleotides long. Many primers is designed for that. This is the genomes that's not required to cut by the restriction endonucleases. That does not require because restriction endonuclease is cost effective. So there is not use of restriction endonuclease. So cost is saving for that techniques. That is the RAPD techniques, and also it is called the polymorphic markers. Why? Because suppose that this is the sequence of the DNA 5 and 3 and 5. So different primers is designed synthetically, that becomes 10 nucleotide longs and randomly mix with that genomic sequences. Okay. So why these primers, suppose that the sequences in that genome is T, C, G. If that complementary sequences is found in that position, suppose that in that position. Also this is a reverse of that primers is identified in that sequences. Sorry, that sequences. Okay. So this is the regions for that's part of segments of that genome. That primer is complementary. Then the synthesis should be occurs. And another, that is many primers used. That's 10 nucleotides long. Okay, 10 nucleotides long. So here one primer is complementary to that, but another is not complementary. In that another conditions, another primer is used that is complementary to here and also complementary to here. That is another regions of the sequences. Now I am marking only for understanding, not it is a cut of that sequences. Only marking for primer if complementations occur of that sequences. Okay. So the result should be that's required PCR. Okay. That's techniques RAPD requires PCR. After PCR, you can also go to the another lecture about the polymerase chain reactions. That's how this process takes place. Then you can easily understand about that techniques. Without knowing PCR, you cannot understand. So PCR amplify a number of sequences, a number amplification that's amplify that sequences. A small amount of DNA is creating or amplifying number of sequences of that specific position by using PCR. Okay, so you can go to the another lectures. That's after PCR, that's amplification regions is like looking number of force in one cycle, that is one cycle. After one cycle, that is renaturations process takes place in the PCR. After denaturations, the double stranded becomes single stranded and the primers binds. After end links, the renaturation process extensions of the DNA at 72 Celsius of the temperatures, the primers extended and amplify that specific positions from forward to backwards. So that is the one cycles, denaturation and extension, and also that specific position amplify as looking like double stranded and also this occurs, but there is not primers. So and also take the examples that amplify the number of sequences. Okay, 4 in one cycle, that is the one cycle of the PCR. In the second cycles, what would be the conditions? Also repeating of that condition of denaturation, renaturation and extensions, the number of 4 becomes number of 8. Okay, 8, and they becomes remain as looking like more single stranded is fast, but double stranded that's remain degraded after sometimes degraded, but the numbers of that specific positions of primers complementary is amplified, and so they becomes as like number of 8 after 2 cycles. It's become also number of 8. Okay, number of 8. So when that's the individuals, I have also described that about the restriction fragment length polymorphism are by using restriction and enzymes, different segments found in the UNI of genomic sequences and also different segments that's differ in length after cutting in U. So that was polymorphic, and you can see that these primers binds if the complementary is found in that position. Suppose that's this is the any numbers of 50 numbers of the sequences, this is 100 numbers of sequences, and for example this is 150 numbers, but these sequences can be changed into the another individuals. Okay, so the sequence that primer cannot bind to that specific position, they can be in another individual as like here and so as like here. That's differ from individual to individuals. So that's primer bind not here and that's binds in here positions, and also they can be changes. So the length of that product would be changes in another individuals. After PCRs, that's the sequences of amplification after by PCRs run this on by jelly electrophoresis, agar jelly electrophoresis to identify the sequences. If that's there is the bands is obtained in the agar jelly electrophoresis, that's bands is obtained. Suppose that this is 5 kb, that is 2 kb in for our genome sequences, for my genome sequences. Okay, after binding to that primers before I have described, but in another individuals for you I am taking the genomic sequences for you, genomic DNA of you, then the primers bind to another positions and the fragments would be differs. That's after jelly electrophoresis that should be obtained 6 kb and 2 kb. Why? Because primer bands attached are complementary to another specific position. So the length of amplification after PCR is differ. So after random amplified polymorphic DNA sequences is found that's differ from individual to individuals, that is called the polymorphic molecular markers, because that's why you can see here. Okay, after electrophoresis, jelly electrophoresis, you can found the differences in the bands. Okay, so this was the examples of the random amplified polymorphic DNA sequences. That's you can know there is the not use of restriction endonucleases, and that's require PCR for amplifications randomly, and also use tag polymerase in the PCR, that is costly, but they can be reused for again amplification of the in the PCRs. So that is cost effective. There is no need of cost in comparison to the restriction fragment length polymorphism, but it is not highly reproducible in comparison to the RFLP. Why? Because the primers can be randomly attached or not attached, so they cannot be highly reproducible. Okay, so that's it can also be it can also be used to map the genomic sequences, but this is a dominant markers. Why called dominant markers? Suppose that this is the mine sequences, and so in another individuals that sequences is not found by the RAPD. That primer does not have any complementary sequences, so there is no band obtained by the RAPD. But after crossing, that is the female, suppose that this is the female, this is the male. After crossing, a new progeny will form. New progeny will obtain. That's genomic sequence becomes isolated of that individual, that is the F1. Okay, that's individual after crossing. Their isolation of the genomic sequences using RAPD, that's required after isolations of the genomic sequences used by the PCR and RAPD is completed, then the bands will be not differ to that is mine. That's also in that progeny will obtain look like 5 kb and also 2 kb. That's recessive condition of the genes does not appear in the F1 progeny. Okay, so that's only show the these individuals in the next generation. So why called this is the dominant markers? In the co-dominant markers, that for RFLP restriction fragment length polymorphism, both, that is 5 kb, 2 kb, and so another that is also 1 kb, suppose that 1 kb and 0.5 kb seen, then the next individual obtain both 1 kb, suppose that's 1 kb, this is 1 kb, also look, and so 0.5 kb also seen in the next generation. So this condition is called the co-dominant markers, but RAPD is the dominant markers. That's you can know about the RAPD. In the next lecture, I would be describe about the restriction fragment length polymorphism and also different types of the RAPDs and SSR, STS, and the different molecular markers that is come to the examinations, and that becomes most puzzle to the students to understanding about the molecular markers. Okay, so thanks for watching, and keep watching this all video of the JK Biology, and share for your friends to that lecture if you like these lectures. Thank you.